I'm CEO of Annexin Pharmaceuticals. We're listed on Nasdaq's First Growth. We're 4 people, key people, I guess, in Stockholm. Then we have consultants all over the world, key physicians that advise us on how to move forward. But we're basically outsourcing the work we do. Therapeutic areas, telling you that we are in therapeutics. We're not doing service diagnostics or anything else. We're developing drugs, or particularly one drug. We decided that it has a good chance to succeed in ophthalmology, which is the word for basically eye diseases, and in oncology. Our main effort is in ophthalmology, in a specific disease that I will tell you more about later on. NXV is the name of our drug candidate. It's provided in a frozen form, in a liquid, transparent liquid. It contains human annexin A5, a protein that all of us are full of. We have it particularly in vascular cells and in barrier cells of different kinds in the body, but every cell produces its own annexin A5. We make a mimic of that we call an NXV. We deliver it via short-term infusion into a vein on the hand, maybe in the arm, with a tiny needle. We may go to a short-term injection with a bolus injection later on. It targets a lipid, phospholipid called phosphatidylserine, or PS, which is lipid that should be on the inside of cells. But when cells are stressed, about to die, or get hurt, physically hurt, they actually flip the PS to the outside. Annexin is there to bind and modulate the activity of PS. If PS is not controlled, PS will interact with other cells to start making aggregates. They will be also cofactor for coagulation. It will be a driver of inflammation. So it's a very basic fundamental biology we're talking about. So annexin is in the body to try to mitigate some of the actions of PS. But sometimes the body's own annexin A5 cannot do it. So we are developing NXV to help improve that situation. In summary, our drug is unique. There's nothing like it on the market or developed by anyone else apart from one company in China. We have a very novel mechanism of action. Nobody else is trying to address that in any similar way. We have proof of concept data from patients in a study in the US, which shows safety and tolerability, which we're really happy about. And we have promising signals of effect. We're working like crazy, I would like to say, to close the database and present some more data after closure of database. We have announced top-line data. And we're basically focusing a lot on finding a partner. Next phase study is likely to be a phase 2B study, maybe a combination of phase 2B and phase 3 study, but we need the finance to do that, and that's best done with a healthy and wealthy partner. We're also building some activity in oncology, which is we'll talk about briefly. So this happens to some patients, or they were well off. They can happen to anyone, typically 50 plus. All of a sudden, the vision is lost. You cannot see sharply. Maybe you can see sharp, but you have areas of blurriness. That can get well by itself in a few cases, but normally it doesn't get well, or it gets worse, much worse, like the image you see on the right side, which is basically can turn into complete blindness on the eye affected. The eye is a very interesting, beautiful organ. Anyways, the light comes through the lens and hits the back of the eye, an area called the retina, or näthinnan in Swedish. There's a little part called the fovea in the middle, where there's very few disturbing things and very high density of cods and rods and cods, basically where you see sharply, right? If you remember from school, that's where you focus when you look at somebody like that. You will use that area, specific area, which is about 1.5 millimeter in diameter, to see sharply. Like any organ, the retina needs blood supply coming from arteries, tiny arteries, in the eye, and is drained by veins. And when you get an occlusion in the venous drainage system, you get like a backflow, and that backflow generates ischemia, meaning lack of oxygen to the cells. Lack of oxygen would trigger production of a substance called VEGF that's trying to get more blood to the eye, but it also creates more edema or more swelling. So you can see the layers of the retina over here. I can't really see it, but anyway, this is a normal retina, but when you get fluid between the layers of the retina, it looks like this. And of course, this disturbs the possibility to look sharp, see sharp. And you get small bleedings and cell death and all sorts of problems. And obviously, vision is a big problem. It's currently treated like illustrated to the left. You get an injection of anti-VEGF, meaning that you treat the swelling by injecting anti-VEGF. You have to do it very often, like every month, normally in the first couple of months. Then you can maybe slow down a little bit, do it every second month. And it looks kind of nasty. Patient gets used to it, but it's still not a very favorable thing to be experiencing. Again, 5 injections normally in the first 6 months. That's a key thing to remember. And this is the $20 billion market, some analysts say. So it's a huge market, even if you have probably never heard of the disease before. Our offer is to do an intravenous infusion in the superficial vein, and then probably also target the cause of the disease and other mechanisms involved in the disease, and a disease-modifying effect. That's what we think we can do. We can change the course of the disease. The patient do not have to get treated every month. And the key mechanism difference between NXV and anti-VEGFs is that anti-VEGFs reduce the swelling. It's supposed to be used for the swelling associated with retinal vein occlusion or diabetic retinopathy. In our case, we can address the stickiness of the blood cells that cause the aggregation and the occlusion in the first place. We are in the anti-inflammatory. We can protect the cells from dying, etc. So a totally different mechanism. That's as a key message for our partnering discussions. So we went to the FDA and discussed with them. We decided to do a phase 2A proof of concept study in the US. It's a very busy slide, I know. 3 doses, looking at safety and tolerability first and foremost. Then, of course, as you can see on the bottom, we look at the visual acuity board when you stand on a distance, trying to see as many letters as you can. We look at the vascular structure in the back of the eye, in the retina. We look at the swelling of the retina. We look at broader areas than just the fovea in terms of function. We look at how many numbers of anti-VEGF injections these patients would get. And we look at the PS target, which is actually circulating in blood as well. So we want to measure PS in blood as a proxy for showing that we have reduced the PS in the eye as well. Key message here is that we have had 16 patients included. We had 1 placebo. Then we got advice from experts in the US saying, well, they saw the safety data from the first 5 patients. They say you shouldn't have placebo anymore. You should do only active treatment. We have 15 treated. 1 got too low dose for technical reasons. 14 patients completed the 4 months follow-up. 12 patients showed improvements or stability in the disease, and 7 of those 12 patients did not require an anti-VEGF as deemed by the eye doctor, while 5 of them needed one injection during this first 4 months, in contrast to normally 5 injections in the first 6 months. So while not a placebo-controlled study, it's quite encouraging. Just to give an example, not a busy one, but you can see this is a scale to the left where you can see the number of letters improving after Annexin. They go up. The swelling, CST in the green, goes down. If you look at the black, gray one on top, you can see an area in the middle where you see the vessels come in and out. The area around that is black, meaning that it doesn't get perfused with blood. You see pre-treatment versus 2 weeks, 4 weeks, and 16 weeks looks much better. The other investigation is the flashing of small, little, tiny flashes to the eye. In this larger area, there's the fovea, and you see the first part here where the fovea was affected. The patient doesn't see anything indicated by black or dark red. It improves over time as well. So both the anatomical changes as well as functional changes. And this is the bottom line. You see the swelling that goes down after Annexin treatment. This is what we call our poster child, a very favorable example, of course. But it still has happened a lot of good things to many patients, or they have remained stable, which is also not expected. I'll talk a little brief about cancer. I told you about that PS is exposed to stress, but cancer cells, they seem to be able to, or they are able to expose PS on the outside as a way of hiding from the immune system. If you have a lot of PS on the outside, you actually stop immune cells from attacking you, which is a smart thing. So it's been named, not by us, but by others, as an Achilles heel of cancer, which is not exploited. This prompted us to start thinking about cancer and more publicly, only at the office. And we raised some funding from our major shareholders to start that effort 1.5 years ago. And it has been shown that Annexin A5 is as effective as a checkpoint inhibitor in animal models as the world-leading checkpoint inhibitors, but by another mechanism. It's also possible to attach a chemotherapy drug to Annexin and make it target cancer cells more exclusively and get inside the cells and kill the cells. And it's also obviously an opportunity for diagnostic, which has been partly shown that you can use Annexin to identify a cancer before and after treatment. So this is just one example before I finish. To the left is a cancer cell, or they're all cancer cells, but they're plated onto a plastic surface. They attach, they start growing. This is a triple-negative breast cancer cell line, which is very difficult to kill in vitro, as well as a very difficult cancer to get. And you see the yellow coloring basically reflects the number of cells from 6 to 72 hours. If you give them no treatment, they grow. If you give them Annexin as such, they grow. If you give them the chemotherapy drug we choose to work with, they still grow. But if we attach Annexin to the chemotherapy drug and link them together, they go inside the cells and they apparently kill the cells and stop them from growing further. This is a preliminary experiment, not preliminary, it's an experience that led us to work with cancer models in animals going forward, and we hope to present some data on that work relatively shortly. Okay, that's it. Eye diseases and oncology cancer. That's where we work. Thank you.
Thank you so much. A lot of pictures, but of course only one that really sticks in my brain, which is, did I get it right? So either it's your alternative or injecting something in your eyeball like 5 times on half a year. Mm-hmm. Yeah, I should all watch like Clockwork Orange for your commercials. Yeah. So, can you describe why your candidate may be comparable to checkpoint inhibitors?
Lung cancer. So, well, basically, checkpoint inhibitors addresses the one specific thing on the cancer cell, which is used by cancer cell to inhibit the immune system attacking it. So basically hides. That's the whole basis for checkpoint inhibition, which got the Nobel Prize, etc. And I mean, this study is done by Johns Hopkins in the US and by Stanford. So Annexin binds to PS, and if you bind to PS, you will stop the PS signaling to the immune system. So the same mechanism, just another target. And so it stops signaling to 5 or 6 different immune suppressive targets, which is a very impressive thing for like a protein drug. Normally you have an antibody which binds to 1 immune suppressive target, or maybe 2 biphasic antibodies. But Annexin can inhibit the signaling for 6 different immune suppressive.
Wow, that is fast. I'm no expert, and I also hear a little bit bad, so I have to speak quite slowly for the next one, so I'll follow you. For the retinal occlusion, is there an obvious comorbidity that gives retinal occlusion as a complication?
There has been some associations with sickle cell disease, which is a rare genetic disease, but otherwise it's typically more prevalent in patients with hypertension, diabetes, heart failure, heart disease. But it's not like super clear that it's always, if you have one disease, you will get retinal vein occlusion. No, that's not the case.
Okay, you'll get a sweet question now. Why is Annexin such an attractive investment at this particular stage?
Well, of course, we presented our top-line data, which is, we've always been saying that is going to lead us towards a potential deal. Of course, we're a listed companies. I cannot speak about how far away or how close we are to a deal, but obviously it's an interesting place to be in right now. And the market or the big companies, they do ask for new mechanisms. So they want, in the eye disease, for instance, they've been working with VEGF, anti-VEGFs, and corticosteroids for 20 years. And they're screaming for more new targets. So that's a reason why it's likely that we will get interest from larger companies.
Okay, I also have to flip that question. What is your Achilles heel as a company?
Oh, small. Having to raise funds now and then, which keeps the stock price kind of a bit low. I should not comment on the stock price, I know, but it's always that thing when the people expect a new round of investments, the stock price goes down, so it goes up and down like that. Small companies are vulnerable. We're at the tail end of the dog And then if things goes up, we will get up. If the dog goes down, we will go down first, right? So it's a sensitive area to be in. And we're small and we need funds.
Yeah, and I think it's good to be front about it because, we all know.
We all know.
It's like that. Okay, thank you so much for your presentation.
Thank you.